Our next speaker, I know, is not here. Faye McLeod, though, is going to be presenting virtually, we're hoping. So again, this is the first time we're actually doing this. I think she's on, Faye. I don't know if you can hear us, but Faye McLeod is from Newcastle University. Here she is. She is going to be speaking about looking at disruptions in early neural networks using human fetal tissues. So, Faye, take it away.
Okay, well, first of all, thank you very much for the opportunity to discuss my work with you today. I apologise for not being there in person. I just had my first son a couple of months ago, so I'm just in the ropes of feeding and all kinds of stuff, but in very, very, very happy place. But yes, the research goes on, and as I say, I'm hoping to we'll have a really good discussion here today. So let me now just share my screen with you, and hopefully this is all going to work fine. Right, can you see my slides yet? Maybe blank screen. Anyone? Oh, here we go, fantastic. We're relying now on my internet. Okay, so sharing the slideshow. We've gone right to the end. There we go. Okay, you can all see that okay? Yes. Fantastic, okay. So, as I say, thank you for the opportunity to speak today. I'm going to talk to you about my model system for modelling STXBP1 encephalopathies in human brain-sized cultures. And this will be a follow-on, actually, from the work that I discussed at the Milan meeting in 2023. So I'm going to discuss with you the differences that we've seen now in neuronal circuit breakdowns and transcriptomic shifts. So this work was done through an Epilepsy Research Institute fellowship at Newcastle University, and as of November I'll be employed as an assistant professor actually within the Faculty of Medical Sciences. So I'm hoping to take this work forward and hopefully build up more future collaborations. So just to start with, I'll give you another brief overview of the human brain-sized cultures that we've been developing here in Newcastle. So we're in quite a new, unique position here in Newcastle where we've got access to the human developmental biology resource. So this is a biobank where we can ethically source human developing fetal tissue. And we can access fetal tissue from as early into the second trimester. So we access tissue that are ages between 14 up to 19 post-conception weeks. And the work that I'm going to discuss with you today is primarily from roughly about 14 to 18 post-conception weeks. So what you can do with these cultures is that you can section them and preserve them in organotypic culture for many weeks to months. And in the schematic here, you can see the main structures of the developing human cortex. So we've got the marginal zone on the top here, the cortical plate, which will go on to form the cortex as we now know it in the postnatal nervous system. We also have the subplate, which I'll come back to very soon, the intermediate zone, the subventricular zone, and the ventricular zone down here. And what you can see here is that with these cultures, this is an example of being 2 weeks in culture, where we can actually maintain both neuronal cells and the progenitor cell markers by these array of different markers at the top here. So we can look at the neurons within the cortical plate, the subplate, and the developing neurons that are coming and migrating up through these cellular structures. And also within the deeper layers, look at renal glial cells and other early progenitor cells as well. And what I find most useful and actually quite very exciting about this model system is that it's highly amenable to viral vector transduction. So in this case, what you're seeing is essentially just the result of a viral transduction of AAV9 under a ubiquitous promoter with a GFP tag. And you can see here that we're able to label many cell types such as radioglial cells, cortical plate neurons, subplate neurons, outer cells. And as I say, this is an example of 17 post-conception weeks for about 2 weeks in vitro. You see very similar structures both between 14 up to 18 as well, just that the subplate is a bit smaller. And this work will hopefully be published soon within the Journal of Anatomy. We've just submitted this work, so it's about a further characterization of this model system following the brain paper in 2023. But just so we're on the same point, a quick digression into why the subplate. So the subplate is a transient structure which only exists in utero in humans and forms the scaffold for neocortical maturation. It's much larger and more complex in humans than in rodents. And importantly, it's actually where the first synapses are formed. So you'll actually see synaptic connections both pre and postsynaptically, glutamatergic and GABAergic, within as early as 14 post-conception weeks. And again, this what again makes the structure really interesting is that there's lots and lots of spontaneous activity present. So there are many calcium transients present within this structure, which I'm going to show you very soon. So essentially, it's likely to be one of the earliest sites affected by variants in STXBP1. So I hypothesised that loss of STXBP1 in early development will alter synaptic connectivity and function in the subplate. And this could potentially lead to adaptation and disruption in the organisation of the neuronal networks, resulting potentially in the dysfunction and seizures in early life. So just as a bit of a refresher, previously in 2023, I published work showing that we were knocking down STXBP1, and we chose to knock down due to the fact that most variants lead to haploinsufficiency, whereby 50% of the protein is reduced. And we were able to do that using shRNA or short hairpin RNA. And essentially we were able to successfully knock down STXBP1, but we wanted to first characterise where STXBP1 was, and you can see here within this immunohistochemistry or immunohistology, sorry, is that the STXBP1 in green is highly expressed within the subplate. And what we went on to show was that we saw a divergent regulation of synapses. So essentially, with loss of STXBP1, we saw a reduction in the glutamatergic synapses, which was identified by the presynaptic marker VGLUT1 and the postsynaptic marker HOMER1, as can be seen here in this image. And we also saw an upregulation of the GABAergic innervation, which is also quite interesting. Furthermore, we saw an impairment in vesicle exocytosis. So what this is telling us is that there's clearly a phenotype here at these early stages, but to really understand the full pathology of STXBP1 pathophysiology, it requires more integrative approaches that really extend beyond these single phenotype assays. So what I've been doing over the last couple of years in Newcastle is to look at this in more detail. So I've been using a variety of calcium imaging, transcriptomics, and some targeted protein expression studies to identify the downstream regulators that's potentially involved in this. So first of all, we've been using, looking at the spontaneous neural activity. So you remember that I told you that this area is the subplate. So all of this work now I'm going to show you is predominantly from, well, in the neural activity is from the subplate region. And essentially this region has a high amount of calcium transit present, with both synchronized and asynchronized. So what you're seeing here is just, we can visualize these calcium transients by, again, applying a viral vector under the synapsin promoter to this promoter. So this will be predominantly neurons, using the genetic encoded calcium indicator GCAMP6F. And you can see here that in this control slice, the at 2 weeks, so all of this work has been done after 2 weeks in vitro, you can see quite a highly synchronized activity as shown here in the analysis afterwards. However, when you look in the STXBP1 shRNA conditions, as can be seen here in the video, there seems to be, it's clearly dysregulated. So the frequency might seem a bit similar, but clearly the amplitude of these events are less. It doesn't seem to be firing in the same way. So when you quantify this and you quantify all of the calcium transients together, you can see that there's no change in the overall frequencies of these events, but clearly there's a marked reduction in the amplitude. We went one step further to try and analyse the coherence of these events. So what you can see when you look at the entire slice is these large wave-like structures that come and contribute towards these highly synchronized events here. And you can quantify that essentially. So the number of cells that are contributing towards the wave and the number of cells essentially that are basically within the cell itself, how much of that activity is contributing towards the wave itself. So you can get two values here for the coherence of these events. And what you can see is that the large wave events don't seem to be changing, but the activity within each cell is less within the STXBP1 conditions. And you can separate out this even further, going into the synchronized and the asynchronized events. And essentially what you can see is that when you look at the synchronized events, so those that are involved in the waves, there's no change in the frequency but reduction in the amplitude of the events. And actually when you look at the asynchronized events specifically, there's an increase in their frequency. So essentially the phenotype that we're observing here is predominantly driven by an increase in asynchronized calcium transients within STXBP1 knockdown conditions. And to further characterize what these calcium transients are, we also applied TTX to these events, and under control conditions you can see that, so TTX, is a voltage-gated blocker, basically a voltage-gated sodium channel blocker, so we'll block all action potentials, and you can see that these events clearly are driven by that synaptic release, so therefore we see this complete blockade, and this is the same both in the control and in the knockdown conditions. So essentially STXBP1 alters the synaptically driven spontaneous neuronal network activity. So this was an interesting finding, and we wanted to then correlate these findings with changes that can occur at the transcriptomic level. So it's known that activity can really drive changes at the transcriptomic level, so we wanted to see whether we can see any differences there. So what we did was we took the same control, so in this case it's our scrambled control and our STXBP1 shRNA conditions, and with 6 slices per condition with 3 biological replicates, again at 17 post-conception weeks, we chose to use the nanostring neuropathology panel. So it's essentially a gene expression panel of over 800 genes involved in cell integrity, neuroinflammation, neurotransmission, for example. So many genes that are targeted for neurology and neuronal dysfunction, to see what effects could be going on. Another good thing about this particular panel is that everything's highly controlled, and you get less variability sometimes than you get with both single cell RNA sequencing and others in that sense. So what you can see here when you check the PCA plot or the principal component analysis, you can clearly see a difference in the control and STXBP1 conditions. So this just assesses the variability within our data set, and you can see that in each of these shapes is a different brain region. So this is, so the square is one brain, the triangle is another. So clearly there's a difference occurring following STXBP1 knockdown. And when you try and then identify what are these differentially expressed genes, you can see that essentially when you run this through analysis in this volcano plot, what we see is of those 800 genes, 81 of those are upregulated and 47 of them are downregulated. And to give us reassurance in our model system itself, the one that's the most highly, so the significance here, so this is how significant the change is, and the log fold change just shows that this is up and down regulated, but clearly this is the most significantly downregulated gene that's present within the data set. So that's a good positive control for us. And then when you want to then identify which genes essentially are what's involved in these genes, what do they what's their kind of function, we then ran this through an engineered pathway analysis, and we found that the top canonical pathways that came out were involved in these 4, for example. So we have synaptogenesis signaling pathway, GABA synthesis, release, transport of cations and amino acids, extracellular matrix proteins. But the largest and most significant one that I'll just talk about for the moment is the synaptogenesis signaling pathway. So this is just the heat map to show you the raw expression changes and the genes that are involved in the synaptogenesis signaling pathway. And I want to highlight just right now the epherin signaling here. So a number of epherin proteins come up here, which is of interest. Furthermore, we've got STXBP1, syntaxin binding 1, syntaxin as well. So you can see we obviously have this quite remarked reduction, is consistent in all of the cases. Again, each shape is a different brain, but there is clearly some variability within the genes involved. But what this is telling us is that there's clearly a widespread transcriptional modelling in the presence of STXBP1. So what we wanted to do then was to see, well, okay, can we then see whether there's some of these pathways within the synaptogenesis signaling pathway changing at the protein level to really understand the signaling cascades further. And just to highlight, most of these genes that have been highlighted here and others, which you can see within our recently published paper in experimental neurology, is that they're all highly expressed within the occipital cortex based on the raw nanostring count. So this tells us that they're really important within the early developing cortex. And furthermore, when you look at the top upregulated and downregulated molecules based on their fold change, again, the genes that are coming up are these ephrin proteins. So the ephrin receptors, ephrin A4, 6 and 3 out of interest. And this one here, NR4A2, is actually highly expressed within subplate neurons. So what we wanted to do was to look at these just by, looking at a standard Western blot. And basically the take-home message is that when you look at the 4 genes that are known to be upregulated in the transcriptomics, it's only the ephrin A4 that's actually upregulated within the at the protein level. And each of these lanes represents a different slice. And we obviously then repeated this 3 to 4 times to ensure significance. We then looked at other components of the synaptogenesis signaling pathway. We looked at presynaptic components, including syntaxin 1A, the binding protein for STXBP1. We looked at MUNC13, and we also looked at GluA2. And of these pre- and postsynaptic components, you can see that out of interest, it's actually syntaxin 1A is actually reduced at the protein level, whereas at the transcriptomic level, it's increased. This may be potentially due to a destabilization of the protein. It's worth further investigating, but this was an interesting finding that obviously the genes are not always correlating with what's happening at the translational level. But what we can say is that there's a selective protein level consequences of STXBP1 reduction, highlighting disruptions in nephrin signaling and presynaptic machinery. And then finally, what we wanted to do was just try and see some sort of functional output. So given that we have this theme of ephrin signaling coming through, we wanted to see whether there was any kind of functional relevance of this. So what we did was we took these early developing neurons and assessed essentially their neurite length from the cell body right up to the growth cone end. And what you can see is that there's a clear reduction in the STXBP1 shRNA conditions when we look at the length. And interestingly, the amount of ephrin A4 is also increased at growth cones. And I should highlight the reason why ephrin signaling is of interest. So ephrin signaling is involved in axonal pathfinding and essentially stabilization of neural morphology. And when you have an increase in ephrin A4, that's known to disrupt axonal growth essentially, and sometimes lead to the collapse of the growth cone. So an elevation in ephrin A4 is actually linked to a reduction in neurite length. And indeed, this seems to be what we see in the presence of the STXBP1 conditions. And although we've not done any genetic modification to obviously get a confirmation of this result, you can see that there is a correlation between ephrin A4 levels at growth cones and the neurite length. And this is clearly separated out within this correlation here, which is highly significant. So essentially, we're seeing that STXBP1 deficiency reduces neurite length, drives ephrin A4 accumulation at growth cones as well. So to summarise, what we see is that we've got this human cortical slice models where we're modelling early neuronal development from as early as 14 right up to 19 post-conception weeks, and we can preserve the structure and look at the function within these different regions. So what I showed you today was focusing on the subplate, but we have looked at the activity across all regions of the developing culture, and I've noticed some interesting diversities within each region itself. But today what I've shown you is that there's a clear difference in the network level with the calcium transients with a STXBP1 knockdown, so there's clearly dysregulation. There's a reduction or a change in the transcriptomics, both in efferent signaling, presynaptic machinery, and I didn't talk about the proteins involved in stress response and growth factor signaling. And I just briefly showed you as well the changes that we also observe within the neurites and the high expression of efferent A4 within the growth cones, which is differing with the STXBP1 conditions. And just to finalise the future directions where we can go with this is obviously what I've shown you with efferent A4 is clearly very much a correlation. We would need to establish a cause of this, and the best way to do that would be to knock down or an overexpression or a knockdown to establish the true causal role of efferent A4 within this. We would also like to identify reversible developmental windows using inducible ST1BP1 rescue models, which we're currently developing in the lab. Try and look at links between the transcriptomal changes and the functional circuit defects in terms of the activity and particular signaling cascades. and finally to test targeted therapies in human slice cultures as well. And with that, I'll thank you for your attention and just want to highlight the people who obviously helped with this work, specifically Gavin Clowry, Andrew Trevelyan, and my PhD student Anna Dempsey, the Human Developmental Biology Resource for their donation of the tissue, and also you for your attention. And I'm happy to take any questions.
Hi, Faye, can you hear me?
I can, yes.
Hi, wonderful talk. It's a beautiful system. I was hoping you could just comment on the degree of knockdown of STXBP1 that you're achieving in your system. You know, I had a similar question in the past talk. I think there may be sort of different pathobiological processes when you have sort of a 50% haploinsufficient reduction versus when you get down closer towards knockout of STXBP1. So where are you on that spectrum, and have you looked at that dose responsiveness to your phenotypes?
So when we designed the shRNA constructs, we did have 3 clones, and we chose the one that was about 50% essentially in our knockdown, and we see roughly about 50% in our in the knockdown. Interestingly, our rescue constructs and the knockdowns are slightly different, which we need to work on, so we can maybe look at the dose effect there. But I think it's important to acknowledge that the degree of knockdown could change the phenotype, and it's something that we should look at further.
Thanks.
Are you seeing differences between males and females? Are you paying attention to the sex?
So yes, we do get the sex of the so we do get the karyotype of the tissue afterwards. So we could segregate out male and females, and we haven't looked at that, but it's definitely something we can look at. We because of the N number that we get with the biological replicates, it's the tissue obviously comes when it comes, but we do have the data to look at that. I think it's something we should do.
Thank you.
Faye, thanks so much. This is Ruud from Amsterdam. Congratulations as well.
Oh, thank you.
I have a question about the FNA4. Can you comment on what your hypothesis on the selectivity of FNA4? And could you also think about, is this FNA4 actually present on the membrane? So you show accumulation, but is it functional, you think?
So in terms, okay, I'll answer your first part. So the selectivity, I would say that it's not the only one, for sure. I think these things work in, the efferent signaling is a complicated signaling cascade. You know, you've got attractors and repulsion signaling cascades happening all over the place, and there's no way that it's working alone. So even trying to target that would be quite, complicated, and it was something we tried to do with a drug which was, yeah, it was complicated, but it's worth trying to see what would happen, in terms of, like the phenotypes that we observe. But I would say that it's definitely working in, combination with others within the system, but it's definitely one that it's coming out quite a lot. In terms of the presence, I need to look further. We didn't do super resolution on our cultures, but it would be nice to be able to see whether it is present on the membrane. I would hypothesize that it is. But again, I would have to check. But one of the beautiful things about these slices that we could do some sort of, super resolution analysis on that to be able to confirm that in more detail.
Yeah, great. Thank you.